Now that we've completed all four processing steps, FTU segmentation, feature extraction, spot data generation, and spatial aggregation, we can see that as a result, we have generated three annotation files. These are the same three annotation files that we see in the Human Kidney Atlas V2 collections frozen samples. First, gloms.json from segmentation plugin. It contains segmented glomerular boundaries. After running feature extraction, this same file is expanded to include 72 morphometric features. Second, spots.json from the set of spot generation plugins. It contains the spatial transcriptomics spot coordinates along with cell type composition derived from transcriptomic data. And third, the final aggregated file, gloms.json, is where everything comes together. It integrates glomerular boundaries and morphometric features from the GLOMS annotation with cell type composition from the underlying spots. This last file is the magical one. It's the file we use for all visualization and analysis going forward. Now, instead of using the data we just curated, we're going to switch to the Human Kidney Atlas version 2 collection for visualization and analysis. Why? Because it contains 153 images, which allows for much more advanced cross-slide comparisons. Let's go ahead and open a file in Histomics UI. Once we are in Histomics UI, click on Others and we will see all three annotation files here. Each annotation has an eye icon next to it. Clicking the eye overlays that annotation directly onto the whole slide image. For example, activating GLOMS makes the GLOMS visible and you can zoom in to explore individual structures. Multiple annotations can be enabled at the same time. Just remember, the last annotation you click appears on top. Also note, since these annotations are rendered in the cloud, loading may take a few seconds depending on server response time. We can check this label box and hover over any structure to see the 72 features and molecular profiles. You can click on an FTU to see the cell type proportions and their states along with 72 features, which shows on the left. You can also select multiple elements by pressing Ctrl key and selecting as many as you want. This will aggregate the values. If you want to lots of elements, for example in case of spots, you can click on this little square icon and then select the region on WSI. It will include all the structure that completely falls inside the drawn square. All of this is powered by the final annotation file generated by the Spatial Aggregation plugin. It lets us explore spatial and molecular patterns in tissue at high resolution. By combining whole slide images, Visium spatial transcriptomics data, and functional tissue unit annotations, we can uncover insights into tissue organization, cellular heterogeneity, and structure-function relationships. You can also customize how annotations are visually displayed. Using gloms-new as an example, turn on its eye icon, then click the gear icon. This opens the Edit Annotation panel. Here you can adjust line width, line color, and fill color. Even better, both line and fill colors can be mapped dynamically to feature values. For example, the outline color might shift from light red to dark red based on correlation eosinophilic, while the fill color transitions from light green to dark green based on L1 cell types, podocytes. This creates intuitive, multi-dimensional visualizations that make spatial patterns immediately clear. All styling changes are saved automatically. They are written directly to the annotation metadata in the Fusion database. So when you reopen the slide, your visualization persists, unless you manually reset it. Fusion's interactive visualizations also support hypothesis generation. By plotting graphs and examining correlations between different variables, which we learn in next video.